Good day and welcome to my presentation, Analysis of Bacterial DNA Sequences Submitted by Peruvian Institutions to Public Databases. My name is Camila Castillo and I will be explaining today what we have done with the information retrieved from public databases. So to start, Peru is a mega diverse country. However, this biodiversity has still not been well researched. We believe that there's a certain preference for certain taxa in all domains of life. And in this case, we decided to focus ourselves on bacteria. So for this study, we decided to choose two public databases that are commonly used by bioinformaticians in general, which is the NCBI's nucleotide database and the Patrick database, this one being an exclusive database for pathogenic agents. The aim of this analysis was to describe and characterize which are the interests of Peruvian microbiology. So, to start with the NCBI's nucleotide database, we found 14,488 records in this database that were related to Peruvian institutions. From all these records, bacteria were the most abundant domain in this database, with 70.60% of all the occurrences, which means that among Peruvian institutions and the sequencing information they generate, bacteria is the most well-represented taxa group. This is followed by eukaryota, which is 24.38%, and viruses, which is 3.07%. No sequences from the archaea domain were found, which indicates that no Peruvian institution has stepped up in the archaea research in the country. So, from this data, we decided to specify which bacterial species were the most represented in the NCBI nucleotide database. We found that Pasteurella muntosida is the most represented bacterial species in this database, followed by Neisseria meningitidis and Vibrio pyrimolyticus. In this figure too, we can see the 10 most represented bacterial species. And in the x-axis, we can see the number of sequences that we found from all these species described here. We found that in the NCBI nucleotide database, most of the institutions that uploaded to this database were public institutions, one of them being the National Institute of Health. So when it comes to the Patrick database, we found that there's one bacteria that is the most represented bacteria in this database, which is Mycobacterium tuberculosis. This may have an explanation in that this country has several cases of tuberculosis still, which may be the reason why Mycobacterium tuberculosis is the most represented bacteria here. Yersinia pestis, Shigella sonei, and Staphylococcus aureus all come much later and with much less representation compared to Mycobacterium tuberculosis. We found that in the Patrick database, the Peruvian institution that had most of the records was a private institution. So, our aim in this study was to, as I said before, characterize Peruvian microbiology. And we found that Peruvian microbiology has a strong preference for pathogenic research, according to all the data that has been uploaded. Although Peru has shown an exciting megadiversity, we still, for example, we still don't have research on archaeas in Peru performed by Peruvian institutions. This means that there are environments and ecosystems that Peruvian microbiology has still not researched or more deeply. There is a strong preference for pathogenic research that may be explained because of the country's continuous cases of infectious diseases. However, there may be certain bacterial species that may be of interest for biotechnology, for example. So this is all from me. Thanks for listening, and I hope to hear your questions soon.